Hi everyone and welcome to my channel. This is Biology with Annie. Today I am going to be explaining about recombinant DNA technology. This will be the first video of a two-part series. Recombinant DNA technology is essentially a piece of technology that utilizes biomolecules like restriction enzymes, DNA ligases, and DNA polymerases to enable processes like DNA cloning and genetic engineering. Now, if you're wondering how it helps to recombine DNA fragments from different species, well, the best example I can give you is that of human insulin. Human insulin was the first licensed drug made using recombinant DNA technology. Essentially, what the scientists did was they genetically engineered bacterial cells to generate human insulin by recombining a DNA fragment coding for human insulin with a plasmid. Plasmid is a DNA molecule naturally occurring in bacteria. Linking the human insulin gene with the plasmid allows it to be carried into the bacterial cell and can now multiply inside the cell so that it will be passed on to all the future generations. Now, when you have a large number of bacteria containing this plasmid, you can induce the expression of the gene, resulting in the production of large quantities of insulin. This insulin protein is further processed, purified, and packaged to become the insulin drug. Now, this process involves multiple steps. The first step is to isolate the gene of interest or the insert. Let's say you want to isolate a gene from the human cell. One approach is to use restriction enzymes to cut the gene from the rest of the genome. If you want to know more in detail about restriction enzymes, then I highly recommend you to watch my previous video. I will leave a link for it in the description box below. Now imagine there is no appropriate restriction site around your gene. Then what do you do? An alternative is to chemically synthesize your gene of interest. This involves knowing the sequence of your gene and adding the nucleotides in order one by one via the phosphoramidite reaction. When generating the fragment, restriction site sequences are added at the ends so that they can be digested by restriction enzymes. This will provide the sticky ends needed for ligation. The major drawback of this approach is that the reaction only allows the synthesis of oligonucleotides ranging in size between 10 to 100 bases. Larger fragments tend to have inaccurate sequences. So, if your gene of interest is larger in size, then you will have to try the third approach, that is amplification using PCR. PCR or polymerase chain reaction is a technique that uses chemically synthesized oligonucleotides, also known as primers, and an enzyme called DNA polymerase to direct synthesis of DNA fragments from a template. So, if the genetic material in a human cell is isolated and added to the PCR mixture, DNA polymerase will use it as a template to synthesize copies of it. Which regions of the genome is amplified is determined by the primers. So the primers are generated chemically using phosphoramidite reaction as mentioned earlier. The sequence of the forward primer has to be complementary to the starting sequence of the gene. Similarly, the sequence of the reverse primer has to be complementary to the ending of the gene. Also, as mentioned before, restriction site sequence is incorporated into the primers so that the final PCR products are also containing the restriction site sequence. Once the insert with the sticky ends has been generated, the next step is to digest the vector with the correct restriction enzymes so that the complementary sticky ends will be present on the vector. There are different types of vectors, and the most commonly used one is a plasmid vector. Plasmids are extrachromosomal circular DNA found naturally occurring in bacteria. Hence, they contain the elements needed for replicating in bacterial cells. PBR322 is one of the first plasmids used for DNA cloning. It was named after the two scientists that developed it in 1977, Bolivar and Rodriguez. The number 322 was used to distinguish it from other vectors developed in their laboratory. This vector was artificially constructed by ligating different elements from different naturally occurring plasmids. It contains an origin of replication, an ampicillin resistance gene, tetracycline resistance gene, and about 40 unique restriction sites. Origin of replication is a sequence about 600 bases long. It contains binding elements for proteins involved in DNA replication. This allows the plasmid to replicate using the host machinery. It is important for the vector to have unique restriction sites. This means that in the entire sequence of the plasmid, there should be only one restriction site for a particular restriction enzyme. This will enable us to insert the gene of interest at a defined position in the vector. The third feature a plasmid should have is a selectable marker. In PBR322, ampicillin resistance gene and tetracycline resistance gene act as selectable markers. This enables us to identify which bacterial cells contain the plasmid with the insert. Let's say you want to join your gene of interest with PBR322. You need to digest both the insert and the vector with the correct restriction enzymes so that they are flanked by the corresponding sticky ends. Now, if you add them together in a tube along with an enzyme known as DNA ligase, they will combine to form a recombinant DNA molecule, and this reaction is known as ligation. Now, you need to understand that not all vectors will recombine with the inserts. There is a chance that the vectors could self-ligate despite the non-complementary sticky ends. This is where the selectable markers come into the picture. Once the plasmid is transferred into the bacterial cell, the ampicillin and tetracycline resistance genes helps us to identify the bacterial colony which has the recombinant DNA molecule. I'll be explaining how the plasmid is transferred into the bacterial cell and how to screen for the correct bacterial colony in the next video. So stay tuned. Let me know if you like this video and leave any queries or feedback in the comment section. And don't forget to subscribe to my channel for more videos like this.